Dear students, welcome back to Biology Forum classes once again. This is the only platform where you will get concise, information-rich and concept-based videos. Our classes mainly focus students of higher secondary and NEET examinations, as well as we also cater students from various avenues, competitive examination and higher education as well as. In the previous class, we have studied about the abscisic acid, which is the fifth growth inhibiting hormone in case of plants. Almost we have covered the total hormonal portion or physiological role of each and every hormone in our previous classes. So, quickly get into the recap of our previous class. In the previous class, we have studied about abscisic acid, its bioassay discovery, its physiological role in plants, precursor molecule, site of synthesis and various examples as well. Also, in the previous class, I have concised a table for you about those hormones which both have synergistic effect or antagonistic effect. Hormones having either synergistic or either antagonistic effect in case of plants. Okay? So, in today's class, we will be studying about dormancy, seed dormancy and seed germination procedure. Okay? So, without further ado, let's quickly begin today's class. So, in the previous class, I have already mentioned about two hypothetical hormones which we will study today. The first one is florigen and the second one is vernalin hormone. These are referred as hypothetical or postulated hormones. The actual origin of these hormones or actual site of synthesis of these hormones is not known till today. Okay, but their function or their action is very much prominent in case of plants. And how will they work? Florigen is mainly present in the leaves, young leaves of plant. Okay, and it is meant to focus on the growth of shoot. It is mainly working on shoot apical meristem, and that's why, and this is how they actually work. They actually meant for the growth of shoot of each and every plant. Okay, number one hormone, which is florigen, it is written over here, and the second hormone is vernalin. From the name itself, we can say that it is meant for vernalization or flowering in plants. Okay, it is mainly focused on the vernalization process or the flowering process in case of plants. It is another form of hypothetical hormone present in case of plants. So these are the two hormones which we are about to study in, and I have said already in the previous class that we will focus this on these two topics today. So this much information is enough for your examinations, and this is kind of the question you are going to face in your exam, that what is the function of florigen or what is the function of vernalin in case of plants, or name two hypothetical hormone or name one hypothetical hormone, and like this. Okay, so up to this the hormone part is over. Today we will another part today in our class, the another part is seed dormancy and seed germination. So let's begin that portion. So let's begin with the first portion, which is seed dormancy topic. Okay, so what is seed dormancy? In the previous class, during study of abscisic acid hormone, we have already discussed that the site of synthesis of abscisic acid hormone, and in that particular portion, the first point was dormant seeds. Dormant seed is that, or dormancy of seed is a state where seeds remain inactive in action. They do not germinate. They do not show any kind of physiological reaction going in the particular environment. Okay, simply if we put an if we need to do a definition of seed dormancy, we just have to say that it is a kind of situation in which the seeds remain in hibernated situation or sleeping position. There is no chemical reaction going on, and that is why the seed will not germinate. Okay, so this is known as seed dormancy. Okay, so to study further about seed dormancy, first we need to know the classification of seed dormancy. I think you can see, students, from the, it is written in the board already, the classification. There are several layers in which we can differentiate the seed dormancy accordingly. Okay, the first one you can say, it is written over here, is the pre-dormancy. It is a state where a seed, if a seed will get favorable condition, it will germinate. Pre-dormancy. Pre means before. Before going into the permanent dormant situation, if a seed will get favorable condition, then it will definitely germinate. Okay, this is the first step, which is pre-dormancy. The second step is mid-dormancy. It is the stage where the seed is just going through or a step ahead of getting dormant state. Okay, in case of mid-dormancy, whether you will get or you will give the seed any kind of favorable situation, it never germinates. It remains in that particular dormant state. This is referred to as mid-dormancy state. Okay, up to this I think I can make you clear. Okay, the next portion is post-dormancy. In case of post-dormancy situation, the seed will eventually silently overcome the dormant state and will germinate upon getting favorable situation. Okay, so these are the main three stages of dormancy on the basis of time span. Okay, on the basis of time span, we can say that pre-dormancy, mid-dormancy and post-dormancy. What do you mean by pre-dormancy? I am repeating once again. It is the stage where if any seed get favorable condition, it will germinate. In the next stage, which is mid-dormancy, whether you will give any kind of or each and every type of a favorable condition to the seed, it won't germinate. It will never germinate. It will remain in the dormant state already. Okay. In the post-dormancy situation, eventually with time, silently the seed will overcome the dormant state and upon getting favorable condition, it will definitely germinate. But it will take some time to get that particular favorable condition. Okay, this is the three stages based on the time span. Now coming to the next part, which is innate dormancy. What do you mean by innate? Innate means a situation already is there within itself. If there is higher concentration of abscisic acid present within the seed. So in the previous class, I think you all students can recall that gibberellin and abscisic acid is antagonistic to each other. If in the seed there is higher amount of abscisic acid, then the gibberellin, then the seed will remain in dormant state. It won't germinate at all. Okay, but if it is the opposite scenario, the level of gibberellin is higher and the level of abscisic acid is lower, then definitely the seed will get germinate, because gibberellin hormone is a hormone for seed germination. We all know. Okay. So in case of innate dormancy, if there is already higher concentration of abscisic acid present within the seed, or if there is any anomaly, any chemical or any physical anomaly related to the embryo within the seed, then the seed will never germinate. So this situation is termed as innate dormancy. Okay, if there is any internal cause already present over there. Okay, now coming to the next point, which is imposed dormancy. In case of imposed dormancy, if the outside environment, what are the environments available or important for seed germination is ample amount of water, higher concentration of oxygen, air, etc. Okay, if the outside environment is not favorable, then the seed will never germinate, and this situation is termed as imposed dormancy. Imposed means what? Imposed means we in our daily life we say right, like this, that you impose the situation on me, that some kind of pressure over the seed, outside in unfavorable condition, it puts a pressure over the seed, then it will termed as imposed dormancy and the seed will never germinate. Okay. And the last one is post harvest dormancy. In case of monocotyledon plants like rice, barley, paddy, okay, in case of these kinds of plants, we know that the seeds we collect and stored in the cold storage situation, and in that particular situation, before harvesting, the seeds will remain in a particular we can say dormant state. Okay, so in case of post harvest dormancy, some seeds will remain in dormant state after getting collected from the field. So we can say it like this, that after getting collected from the field, some seeds will remain in dormant situation, and this is termed as post harvest dormancy. So this is a schematic diagram of different classifications of seed dormancy, we can say. Okay, next come to the point, which is quiescent seed. What do you mean by quiescent seed? Before germination, the seed mainly remain in a very dry situation, and in that particular dry situation, it won't get any kind of oxygen, any kind of water from the environment, and that is why all the metabolic activities within the seed will remain in a stop position, and that is why it will not germinate, and this is termed as, this particular state termed as quiescent seed. Okay, so quiescent seed are those kinds of seeds which before harvesting present in a dry, very non-humid environment, and that is why all the metabolic activities within the seed will remain in a stop position. It won't happen actually, and that is why upon getting water and upon getting favorable condition, it will again germinate, and these particular kind of seeds we can say quiescent seed. Okay, I think up to this point all of you can understand what I am trying to deliver. Okay, if there is still any question, you can always comment down below, you all know that. Okay, so next, come to the next another portion of the part is, what are the different reasons of dormancy, why the seeds undergo that dormant state. Okay, so let's continue. So the next part of the class of the topic is the reasons behind seed dormancy, why the seeds remain in dormant state. So you can see over here, it is written over here, these are the various reasons why the seeds getting dormant. The first is water impermeable to seed coat. In case of Fabaceae and Solanaceae family plants, the plants which belong to these two families, the seed coat outside the seed is so much hard that water is really difficult to getting absorbed. What is the point from which water getting absorbed by the seeds? It is termed as micropyle. Micropyle is the portion from which is a you can say it is a pore by which seeds absorb water from the environment, and the water will get entry into the seed coat, and it will start the metabolic activities, and the seeds eventually germinate. But in case of the Fabaceae family and in case of Solanaceae family seeds, their seed coat is too much thick or too much hard that the observed water is not getting efficiently absorbed, and that is why the seeds will remain in dormant state. Okay, the second point is, if the seed coat is impermeable to oxygen. Okay, in case of various weeds and sunflower tree, the seed coat is too much tough that oxygen is the oxygen facility is not good there, or they are impermeable to oxygen, and that is why upon getting low amount of oxygen, the respiration procedure will disrupt, and that is why the seeds won't germinate. Okay. The third point is immaturity of embryo. In case of ginkgo biloba, or in case of various orchids, you can say that the embryo within the seed remain immature. That is why upon getting detached from the tree and upon falling into the soil, because of the immaturity of the embryo, the seeds will never germinate, or it will take some time to recover from the dormant state. This is the third point why dormancy will occur in the seeds. Okay. The fourth point is mechanically resistant seeds. In case of amaranthus, in case of capsula, you can see it is written over here. In case of those plants, the seed coat is too much tough, or too much thick, that the seeds will remain in quiescent or in dormant state, and it will never germinate, or it will take very big amount of time to germinate. Okay. The fifth point is necessity of low temperature. We all know that apple grows in Kashmir, and the temperature of that particular region is very much low, 5 to 10 degrees almost. Okay. In case of peach also. So in case of apple, in case of peach, these kinds of fruits which mainly harvested in colder areas of our country, the necessity of low temperature is very much important. If there is not sufficient amount of low temperature, the seeds of those plants will never germinate. They must need that amount of low temperature to get germinate. Okay. The sixth point is influence of light. Now comes to the next point is lettuce and tobacco are those plants we can say positively photoblastic. What do you mean by positively photoblastic? Photoblastic are those plants of the seeds of which plants need light to germinate. Okay, this is called photoblastic plants. The seeds need light, ample amount of sunlight to get germinate. Tobacco and lettuce are two examples of that kind of plants which the seeds of which plants will get germinate when there is ample amount of light. That is why they are termed as positively photoblastic. The opposite scenario in case of onion seeds or in case of nigella seeds. Okay, which we actually the seeds are black in color, and from this we can make a reminder in our mind that the black seeds means they do not need any kind of light to get germinate. So these nigella seeds or onion seeds are called as negatively photoblastic. Negative means not light, no requirement of any light. So that is why they don't need any light to get germinate. Which seeds? Onion seeds or nigella seeds we can say. Okay, so these are the various reasons of seed dormancy. The first one is water impermeable through seed coat. The seed coat is too much thick that water is not able to get inside the seed and to start the metabolic processes. That is why the seeds will remain in dormant state. The second point is oxygen. There is less amount of oxygen. The seed coat is impermeable to oxygen absorption, and that is why the respiration will disrupt, and that is why the seeds will never germinate. Okay. This kind of scenario we can see in case of various wheat and sunflower. The third is immaturity of embryo. Upon getting detached from the tree and fall into the soil, the seeds are not be able to germinate because the embryo within the seed is remaining immature. Until it will attain a state of maturity, it won't germinate. Okay. In case of ginkgo biloba, in case of orchids, we can say this kind. We can see this kind of dormancy. Okay. Next, mechanically resistant seeds. The seeds of capsula, the seeds of amaranthus are mechanically so much hard, so much tough that it won't germinate. Necessity of low temperature. The fruits which grow in winter countries, winter areas of our countries, apple, peach, they need that time of low temperature to get germinate. Okay. And the last point is influence of light. In case of nigella seeds, in case of onion seeds, they are black in color and they do not need any light to get germinate. That is why they are called negatively photoblastic. On contrast, Lettuce and tobacco are positively photoplastic because they must need ample amount of sunlight to get germinate. Okay, so these are the various reasons behind seed dormancy. Okay, let's continue with the next portion. So this is the next part of this topic, various artificial methods to break seed dormancy. For agricultural purpose, we need very short span of time for seed germination. Okay, so these are the various artificial methods by taking which we can make the seeds possible to germinate. Okay, the first is scarification. Scarification, scar means any kind of wound, any kind of mark in our body. Okay, scarification means we make the seed able to germinate by putting external mechanical or chemical pressure. Okay, you can see over here that scarification is classified into two types. First one is mechanical and the second one is chemical. In case of mechanical, we rub or we hit the seed, that particular type of seeds, which seed coats are very thick or very hard to crack. Okay, so we put pressure from outside environment by hitting the seed, by rubbing the seed over hard surfaces, or by rubbing the seed by using sandpaper. You know all the sandpaper is used for painting our home, painting our outside interior of our house. During the particular painting situation, the painter rub the sandpaper over the wall to make the surface smooth enough for by which the paint will actually absorbed easily, can put the paint easily in the wall. Okay, on the wall. Okay, so by using that particular sandpaper, if we rub the seed coat, it will eventually disrupt and the seed will eventually germinate. So this is the mechanical process by which scarification will be done. Okay, the next is chemical. If we soak that particular seed, hardly or thick seed coated seeds in sulfuric acid like H2SO4 or hydrochloric acid like HCl, if we soak the seeds within that particular high amount of acidic content, then the seed coat will eventually melt and the seed will be open for germination. Okay, these are the two methods by which scarification will be done. The next is application of pressure. So in case of alfalfa or clover plants, in case of alfalfa or in case of clover, at 18 degrees Celsius and 2000 atmospheric pressure, if we put over the seeds, upon 15 to 20 minutes the seed will start germinating. Understood? In case of alfalfa and in case of clover plants, if we put 18 degrees Celsius on 18 degrees Celsius temperature and application of 2000 atmospheric pressure, upon 15 to 20 minutes or 5 to 20 minutes, the seed will eventually start getting germinated. Okay, so this is another artificial procedure we can follow for seed germinate, for breaking seed dormancy. Okay, the next point is low temperature. This particular point, we can say another name is stratification. What? Stratification. The first one would be scarification and the application of low temperature. If we keep the seeds coated by sphagnum, one kind of moss named as sphagnum, and keep it in 5 to 10 degrees Celsius temperature, which is termed as stratification procedure, then the seeds eventually start getting germinating. Understood? If we keep the seeds coated by a moss named sphagnum. What is the name of the moss? Sphagnum. The name of the moss by which we coat the seed or cover the seed is named as sphagnum, and keep it in 5 to 10 degrees Celsius temperature, very low temperature, then eventually the seeds will start getting germinated. Okay? Coming to the next point, alternating temperature. If we rise the temperature and lower the temperature or in alternative pattern, then the temperature difference will make the seeds to get germinate. Okay. The next point is light. The photoplastic, in case of photoplastic plants, if we put white light and red light alternatively, then this light difference or this shifting of red to white light will eventually put a pressure over the seeds and the seeds will get germinate. Okay. The next portion is hormonal application or hormone application. We know all that cytokinin hormone is meant for cell division. Gibberellin hormone is meant for seed germination. If we put artificially these hormones from the outside to the seeds which are unable to getting germinate or remain in dormant state, then upon application of this type of particular hormones, the seeds will definitely get germinate. These are various artificial methods by which we can break the dormancy state of any seed and make them germinate. So coming to the next part and the last part of this topic is the importance of seed dormancy. The importance of seed dormancy is that whenever there is unfavorable condition outside, we can make them possible to germinate by application of this type of artificial methods. And seed dormancy also prevent viviparous seed germination, as well as the seeds which we consume for our protein enrichment or nutritional enrichment like flax seed, chia seed, sunflower seeds, okay, or pumpkin seeds, we can say. These seeds remain in dormant state, and that is why we are able to consume these for our better source of, as a better source of protein or better source of fat. Okay, so these are various importance of seed dormancy, we can say. I think up to this I am able to make you understand the particular topic. Our next portion is seed germination procedure, which we will study right now. So let's continue with the next part, which is seed germination. Okay. Okay students, let's continue with the next portion of our today's class, which is, you can see it is written over here, which is seed germination. What is seed germination? Just now we have studied about seed dormancy is that a particular state of a seed or particular condition of a seed in which it is in sleeping condition or hibernating condition. It won't germinate. But upon getting proper environment which is favorable to the seed germination, they eventually definitely will germinate. So to study about the seed germination, we can say that, for example, if this is a seed and outside of the seed is a seed coat present. What is this? Seed coat. And this is a viable seed, means living. Okay, this is a seed, this is the seed portion, and the outer portion, the covering outside the seed, is termed as seed coat. And just here is a pore present which is named as micropyle. From that particular micropyle, water will get entry into the seed from outside environment. Hope I think you can make you understand. Think that this is a medium where the seed is kept, and the outside of the seed is water present, and from that particular pore present in the seed, which is termed as micropyle, water will get entry into the seed. Sorry, this is not here. This is here, micropyle. This is the micropyle portion. Okay, water will get entry into the seed coat, and eventually the seed coat will swell up, and the seed will get enough amount of water to start its metabolic activity for germination. Understood up to this? This is a seed, outside covering is seed coat. This is a live, or this is a viable seed or live seed. This is the portion termed as micropyle, through which from outside environment continuously water will be soaked up by the seed, and the seed will swell up. Okay, this process, this swelling of seed, is termed as imbibition. imbibition. Okay, and the imbibition procedure will supply sufficient amount of water to the embryo present within the seed, and the embryo will eventually get mature. Okay, up to this point I think I can make you understand about the procedure. Okay, so coming to the next part. In case of dicotyledon seeds, if this is a seed, these are the cotyledon or cotyledon, and the upper portion of the cotyledon, this is the embryo axis. This is the embryo axis. Okay, and the upper portion of embryo axis by which the cotyledon is attached is known as epicotyle. So this is a picture of dicotyledon seed. These two are cotyledons in case of dicotyledon seed, because di means two, and in case of monocotyledon seed only one cotyledon will be there. This is the embryo axis. This is the embryo axis. The portion of cotyledon which is attached to the upper portion of the embryo axis will be termed as epicotyle, and the lower portion of cotyledon which is attached to the embryo axis will be termed as hypocotyle. From the hypocotyle, radicle will be formed, and from the radicle, primary root will be formed. From the epicotyle, plumule will be formed, and from the plumule, shoot will be formed. Okay. Let me add an additional information here that radicle from hypocotyle portion will be formed first, and then plumule from the epicotyle portion will be formed. First radicle will be formed, and next to that plumule will be formed from epicotyle portion. Okay. Any other root formed from radicle, any root formed from radicle will be termed as primary root, and any other roots which will not form from radicle will be termed as adventitious root. Okay, I think up to this I am able to make you understand about the total germination scenario, the total pictorial representation. Okay, let's coming to the next part, which is the different types of germination. So there is different types of germination, we can say epigeal, hypogeal and viviparous germination. The first one is epigeal germination. Epigeal means the first development of hypocotyle. From epi we definitely first connect that epigeal means the formation of epicotyle first, but here the scenario is just opposite. Epigeal means first development of hypocotyle, and hypogeal means just opposite, first development of epicotyle. Okay, we all know that from hypocotyle root, mainly primary root is formed, and from hypocotyle primary root is formed, and from epicotyle shoot will be formed. Okay, so in case of epigeal germination, first hypocotyle will develop. Okay, what kind of germination is this? Suppose this is a seed. Suppose this is a seed, and from that plumule, and this is the portion from which epicotyle means plumule and hypocotyle means radicle will be formed. This is a seed, and this is the epicotyle portion from which plumule will be formed, and this is the hypocotyle portion from which radicle will be formed. And upon getting favorable condition, upon getting ample amount of oxygen, nutrient, water, etc., total favorable condition, it will start germinating. And in case of epigeal germination, the developing seed, in case of epigeal germination, first develop what? Hypocotyle. The hypocotyle will develop first, and it will eventually push the epicotyle portion and the cotyledon above the soil level. If this is the soil level, the development of hypogeal portion, the development of hypocotyle portion, the development of radicle, the development of primary root will eventually push the total seed cotyledon and epicotyle portion above the soil level, and this type of germination will be termed as epigeal germination procedure. Okay, I think up to this you have well understood the portion. Okay, in the next part, which is hypogeal transformation. So let's continue with that. In case of hypogeal, the scenario is just opposite. It is the first formation of epicotyle. First, what will develop? First develop epicotyle. Just remember it with a trick that epi means epi to hypo. The coordination epicotyle, hypogeal germination, hypocotyle, epigeal germination. Okay, just make it remind like this. So, in case of hypogeal germination procedure, the first development will be done in epicotyle portion. The scenario here is like that. If this is a seed and this is the epicotyle and this is the hypocotyle, in case of the next step, if this is a soil level, sorry, at first the seed will be inside the soil, and from this the epicotyle portion will get developed and the hypocotyle portion will not. Okay, and this total portion is termed as hypogeal transformation, or sorry, hypogeal germination. In case of hypogeal germination, the epicotyle first will develop, and in case of particular scenario, the seed, from that seed, the hypocotyle, not the hypocotyle but the epicotyle will get develop into plumule first and then eventually into the shoot. In case of pea, in case of mango, in case of wheat, in case of rice, this particular type of germination procedure we can notice. In case of pea, in case of mango, in case of rice, mainly the monocotyledon plants. So these are the examples of hypogeal and epigeal germination. In case of epigeal germination, mustard, bean are the examples, and in case of hypogeal germination, wheat, mango, rice, okay, the pea, these are the examples of plants. Okay, let's come to the next part, which is viviparous germination. It is seen in Sundarbans region or Sundarban reserve forest. When the seed will remain inside the fruit and attached to the mother plant, and in that particular situation the seed start germinating, then it will be termed as viviparous germination. Upon getting developed within the fruit itself, the seed will start germinating and the hypocotyle portion will eventually grow. And when the seed with the fruit will detach from the mother plant and just put in the soil, then from that particular portion a new tree, a new life will get formed. Okay, this is the viviparous germination procedure, we can say, which we can consider or we can find in Sundarban reserve. So up to this point, I think I have covered the total portion of seed germination procedure with their various types. If there is any question still remain in your mind, you can mail me in my email ID for further clarification, or you can comment down below for further questionnaire if there is any. Okay. Okay, so up to this point I think I have covered the total seed dormancy and seed germination procedure. If there is any question from your side left, you can also comment down below for further clarification, or you can mail me in my email ID which is right now popping in the screen for further any kind of questionnaire. Okay, so if you like the video and if you enjoy the learning process with me, don't forget to share the video as much as possible, subscribe to this channel and put the notification bell on always, so that every time I upload any video you will be get notified. So thank you so much.